Kavita said, I am also by background a physicist, though I haven't really done physics for a while now. And I'm not a population biologist or an evolutionary biologist, but I got interested in an evolutionary problem relating to what I mainly work on, which is transcriptional regulation. So it's unfinished and unpublished work, but I thought I'll inflict it on this audience. TFBS is transcription factor binding sites. And for those of you who are not that familiar with what goes on when genes are transcribed, transcription is the process of turning the gene, the DNA material, into an RNA molecule, which is then translated somewhere else into a protein. And there are these proteins called transcription factors that come and bind to the DNA to sort of tell the system when the gene is supposed to be turned on, or maybe when it is not supposed to be turned on. This guy is meant to be the RNA polymerase, which is the machine that actually makes the RNA molecule. The region immediately upstream of the gene is called the promoter region, and a bunch of transcription factors typically bind there. There are other places further upstream where also they could bind, which could be in sequence space quite far away from the gene, but because DNA is a 3D object, they can still interact with the promoter complex. There are other things going on, like methylation in the DNA can affect binding of proteins. There are things called insulators that if this binding factor binds to an insulator, then it blocks the effect of this on this. So it's a very complicated problem. So what I'll basically talk about is transcription factors, what they bind to, and what are the selective pressures on the sequences they bind to. So transcription factors are those proteins. They bind to short sequences, 6 to 20 base pairs. I'm sorry. We'll come back. Okay. Yeah. So you'll often see in papers binding motifs are represented like this, which visually sort of means that a T is likely at this position and a C is likely at this one, and these positions are not terribly informative. These are basically translations of the usual description of a binding site, which is a position weight matrix, which will come to, into a visual representation. It's a one-to-one mapping. And now the conservation and the turnover, the evolution of TFBS is a long-standing problem of interest, because it's widely believed that, so it's come up here too, that a lot of action in terms of variation in populations and in evolution occurs not at the gene level, but at the regulatory level. And so understanding these things is very much an important research topic. So what is the position weight matrix? Well, in mathematical terms, it's basically an assumption that every position within a binding sequence is given independently by a multinomial distribution over the 4 nucleotides. So if you're given n binding sequences, each of the same length L, what you're doing is you're assuming that each column is independently sampled from some probability distribution. And then given these sequences, the probability of seeing a nucleotide alpha, where alpha is A, C, G, or T, in one of the positions, I mean, a new binding sequence is this, where big N is the number of sequences you have in your training data. Small n is the number of sequences in your training data where your nucleotide occurs at the position of interest. And these things are called pseudo counts, and they can be formalized via using a Dirichlet prior and so on. So for those of you who are familiar with it, C is equal to 1 represents complete ignorance of the prior distribution, and that's basically Laplace's rule of succession. In practice, C is half is often a good choice, and I won't get into why, but there are theoretical arguments why you should use half. And the sequence logo that we showed earlier is a representation of that matrix, where each letter is represented in proportion to its abundance. So A and G are the most abundant here, so A is represented slightly higher than G, and a little bit of T there. The total height is the information score, which in this case is defined like this. Most of you might recognize information score as minus of this term, which gives you a maximum when all possibilities are equal. But it gives you a minimum when you have one of these is 1 and the rest are 0. We want exactly the opposite of this. We want a strong signal when you always see A, which in the Shannon term sense is least informative. And we want a minimum signal when they're all equal, so we plot it like this. And this is kind of it was introduced, I think, in the 90s sometime, and it's become, yeah? Yes, the minus of this is exactly the Shannon entropy. It has a minimum value of 0 minus W log W is minimum 0 when when one of them is always there. For example, so there are four numbers, each of which has a probability between 1/4 and 1, and they add to 1. So if all of them are 1/4, you'll get the maximum value for this, which will be 2. And if one of them is 1 and the rest are 0, then you get a 0. So basically, to invert that, it is 2 minus the Shannon entropy. So the evolutionary questions that people are interested in are, first of all, mutations inside the binding site can affect gene expression, therefore fitness. But and species are distinguished at least as much by regulatory differences as by differences in gene expression. But it's not like you mutate a TFBS, immediately something happens, because especially in metazoa and so on, you'll have lots of redundant binding sites. And there is turnover, there is compensatory mutation, all kinds of complicated things happen. So relating TFBS mutations to fitness is really a complicated problem. There are people who've been trying to evolve models of gene expression of binding sites within enhancers with dependencies on other binding sites and biophysics and so on. So it's still, it's very early days in that. So we are going to be looking at a slightly simpler question, that apart from the level of individual sites, within an individual site, are the individual nucleotides themselves under selective pressure? And that's, well, obviously the answer is yes, but what kind of pressures are they in? And that's what we'll be talking about here. So the tool we use is the standard general time reversible model of nucleotide evolution. Well, okay, so this is the time reversible model. We'll be using something else. So the idea basically is that each nucleotide can mutate to any other nucleotide. So you have a rate of a mutation matrix, a substitution matrix, a rate matrix, where the off diagonal elements are rates of substitution from beta to alpha. And you define the diagonal elements in this way so that the divergence matrix, so this comes from continuous time Markov chains, and it's a standard textbook derivation. So then you can define a divergence matrix, which is if time t has elapsed between your ancestor nucleotide and the one you observe now, then given the ancestor beta, what is the probability of observing alpha? And that is given by the basically exponential of this matrix. And it's quite easy to derive. And the point is you can have multiple mutations in that time. So though, so if you define this as a substitution rate matrix, purely the diagonal entries will be 0. We define it in this way to basically make the math easier. And it works out. This accounts for new mutations coming into your, thing. And this matrix can have non-zero diagonal entries, which basically means either the alpha never mutated beta never mutated to alpha, or there were compensatory mutations. If the process is time reversible, which we expect it to be, then you will have a stationary, if you have a stationary distribution pi alpha, that means the probability of seeing a given nucleotide alpha in the long time limit is pi alpha. That distribution should be stationary under this process, and that gives you the detailed balance condition, which we'll come to later. Well, so the point is that this is looks like a good starting point. It turned out that it's kind of hard to use. Well, so the general approach is to use this to study this position-specific evolution, take a large collection of sites in one species, like human, and orthologous sites in related species, and then place these on a tree with the branch lengths estimated from neutral sites or from the literature, whatever. And train this divergence matrix with its parameters in a position-specific way to maximize the likelihood of the data that you do see, and then calculate what the stationary distribution would be according to that divergence matrix. And does that stationary distribution, is it in any way similar to the position weight matrix? So basically the, yeah. Yeah, the sequences that we actually have are the leaves, and the nodes are unknown ancestors. It's a snapshot in time. So it's a large collection of sites within human, within chimp, and so on. Right. So the full set of, so we have this independently for each position within a binding site, and so you have a vector of stationary values, and this n by 4 vector forms a matrix like the position weight matrix. But it has a different interpretation. This is a normalized base count within your species of interest, whereas this is the stationary distribution uh, derived from the multi-species data, and you can use a sequence logo to visualize it in exactly the same way. Now you would expect that the PSSV shape, so we'll come to what happens. You expect that shape should be similar to a position weight matrix logo, but if the selection pressure is not terribly strong, then you might, or turnover is significant, then you might have a lower shape, but I mean lower height overall, but a similar shape. So what kind of model do we use? We could use this HKY model, which is basically Jukes-Cantor type thing, but treat transitions and transversions separately, or the general time-reversible model where you treat all substitutions separately, or this specific question of nucleotide evolution was treated by Halpern-Bruno in the context of amino acids. So we never talked about selection. We kind of assumed that selection will be reflected in your equilibrium distribution. What these guys do is they include fitness effects, assuming that fixation, selection fixation occur much faster than mutation. But so they start from this model and they put fixation on top of that, and they end up with something. They end up with a matrix that's similar to what you'd get with GTR, but they have a lot of parameters. So at this point, I haven't tried doing this, but there are others who have tried to do this in transcription factor binding sites, but not really explore the evolutionary questions. More asking whether the distribution of nucleotides you see is what you expect from this model. So the goal is to model it using time-reversible model with position-specific parameters, fixed branch lengths. That is, we are taking a tree as given, and then to calculate the stationary distribution at each position. Now, the GTR model that I mentioned earlier, it turns out if you make the assumption of uniform overall mutation rates, that is, you assume that every nucleotide is equally likely to mutate with a rate mu, and it's only what it mutates to that differs, then it turns out this model cannot reproduce the sort of skewed nucleotide distributions you get in a position weight matrix. So if I have time at the end, I'll just go through a quick proof why. Halpern-Bruno and some more sophisticated models are a kind of, I might still explore it at some future time. But right now, we're going with the Felsenstein model of 1981, which is a much simpler model. It basically says a divergence matrix given a mutation rate and a time t. Either it has not mutated, in that case, the probability of it not mutating is e to the minus mu t times delta alpha beta. Or if it has mutated, it simply assumes that it has sort of fixated to the equilibrium vector. So it doesn't make any sense really at the level of an individual nucleotide. If it, yeah, I'm using F81 here. No, but I wanted to explain the issue with GTR. F81 can be put in the GTR time in the framework if you allow non-zero diagonal elements. Then it becomes the same thing. Yeah. So the idea is that naively you would expect if you mutate a nucleotide, then it has to be different, so it cannot be this. But at the population level, it could be that if you mutate it, either mutation got lost or it got fixated, and therefore the probability of seeing this nucleotide in the population after a while is still pi alpha. So that's at least how I choose to interpret it. I mean, you might disagree, but the main thing is, it's easy to calculate with. So now the one of the things that we'd like to look at is position weight matrices assume that each column is independently sampled from its own multinomial distribution, but in fact it's known that there are correlations within sites, and this is another research instrument of mine that I won't get into now. But what we can do with this approach is we can infer ancestral distributions, and then we can condition the evolutionary model on ancestral nucleotides. We'll come to. And there is a possible explanation for intrasite dependencies that, for example, if you have one of these two positions needs to be the good guy, but if you have a C here, then this one could be a G or a T. But if you don't have a C here, then this one has to be a G. That kind of situation can occur, and that can cause these dependencies even though you're going with the weight matrix model because of the fitness situation. So this tends to support this view that it could happen even within the position weight matrix framework. So here's briefly what we did. 7 primates, 5 yeasts, Saccharomyces yeasts, 5 fruit flies, and the 3 topologies were taken from the literature. Binding sites using position weight matrix from high throughput binding experiments. And the binding sites were required to be conserved across all species, which means no indels within the sites and at least 50% sequence identity. And so this gives results for several factors from each of these. And the first row is the position weight matrix. The second row is this PSSV, which gives you the selective pressures. And you can kind of see that it's mostly pretty reflective of there aren't any major differences. In the case of fruit fly, the overall height is consistently less. There have been papers talking about turnover in fruit fly binding sites that came, I mean, this is some 5 or 7 or 10 years ago. I don't know whether fruit fly turnover is likely to be more than these. It could be that my conditions for choosing weight matrices were more stringent for some reason in human than they are here. You can see some minor differences here, like in MVP1, the PWM prefers a T here slightly, but this one prefers a C here, but it's just a slight difference. And similarly, there is some signal here which is missing here, but there is some signal here that's missing here. And again, so these are things that you can look at, notice when you look carefully. But for the most part, the conclusion seems to be that the selective pressures within a site are quite reflective of the frequencies of all sites within the species. Now we look at the question of compensatory mutations. So supposing, for example, you look at a case where you have an A or a G that is allowed here. And now you can infer likely ancestral nucleotides for each of these sites that you look at in human. So supposing you have 30,000 human sites, well, these are yeast. Okay, you have maybe a few hundred yeast sites, and you have an ancestral, some of them have an ancestral A, some of them have an ancestral G preferred. And if you single out that ancestor, then you actually see these induced effects that if you have an A in this position, you strongly prefer an AT over here, somewhat strongly, and also this T over here. Whereas if you have an ancestral G, then you have a different kind of pattern here, and you prefer these T's here. It's an even more drastic effect with the MXY1 in human, that this guy can be either a G or a C. It's usually a G, but sometimes it's a C. If it turns out, if you pick the ancestral G, then the pattern is quite similar to what you see. But if you pick the ancestral C, then you induce a strong preference for this double C over here. So at this point, I would say that this is a nice tool to look at the effect of compensatory mutations, and as well as to look at selective pressures on individual nucleotides within binding sites, which is an interesting question from the evolutionary point of view. Ideally, of course, so what we have done at this point is use gypsy experiments in human or in Saccharomyces cerevisiae or whatever, and then used conservation and multiple alignments to predict sites in other species. Ideally, you would have actual binding data in other species, and of course, using a better model like Bruno might be eventually more desirable. Since I think I do have enough time, quickly talk about GTR and this question of skewed distributions. So the point is that this stationary distribution pi over here, in typical applications, each pi doesn't vary much from 1/4. It might be 1/4 to 1/3 depending on the nucleotide. Whereas in a weight matrix, you might have pi a is 0.9 and the rest are very small. Now, numerically, I discovered that you cannot reproduce such things. Now, the reason it turns out is something like this. Supposing you supposing you assume a uniform mutation rate mu, which does not depend on the nucleotide, then let's say alpha mutates to beta at different rates in the rate matrix, but the sum of all of these is equal to mu. And then the diagonal elements of R are all equal to mu. And then you can rewrite this divergence matrix in this form, where P is R plus I divided by mu, you can even derive this equation, which I won't do right now, but sorry. You can derive this equation by saying what is the probability. So P essentially is the probability given that a mutation has occurred of seeing alpha where the ancestor was beta. Now, if a finite amount of time has occurred, what is the probability of seeing alpha given beta? You can write that as the probability of seeing this given that no mutation has occurred, or given that one mutation has occurred, which is P, or given that two mutations have occurred, which would be proportional to P squared times the, and each factor weighted by the Poisson distribution. And if you calculate that, you exactly get this formula. And you can also show from this formula that if T satisfies detailed balance, so must P. So basically, that's the interpretation of P. Now, if P satisfies detailed balance, and then the most general matrix that can satisfy detailed balance can be written like this, where A, B, C, D, E, F are arbitrary parameters. And their columns sum to 1 because each nucleotide mutates to something. Now, given this set of constraints, supposing, for example, pi t is greater than half, and pi a, pi c, pi g are each less than 1/6, then you can show from these three equations that C, E, and F are each less than 2, because pi t is greater than half. But if C, E, and F are each less than 2, and pi a, pi c, pi g are each less than 1/6, then this equation cannot be satisfied. So cases where one nucleotide predominates cannot be satisfied in the GTR model if you assume that all mutations are equally likely. So the key assumption here is really that the mutation matrix can be written in this form, where basically the R matrix is basically P minus I, sorry, R matrix can be written as P minus I times a constant mu. Now you can have more general R matrices that do satisfy this, but you just have lots of parameters then. So it's a question that I'm still thinking about. So I'll stop with this. This problem came about in the context of some ongoing work with Lilanti Narlikar, and I've also had nice discussions with these other people. So thank you very much. Yeah. This one? Regular as in? Oh, this one. This is a highly conserved motif. In general, the shorter the motif, the more uniform it would be, because you need each of these to be correct. And so there's a lot of theory behind. Okay, one of the things. So if you look at bacterial motifs, they are often even stronger. So there is some literature on whether it is better to have a few strong motifs or lots of weak. I mean, few strong sites or lots of weak sites. One thing is simply there is not that much intergenic space in yeast. It's less than 1,000 base pairs in most cases. There's lots of intergenic space in Drosophila and even more so in human. So you have space to have lots of different sites. And a lot of Drosophila factors, many of them have homologs, much more so than in yeast. So you have many related factors binding to different sites and sometimes competing for binding to the same site. So things just seem to be blurrier for these reasons. In fact, it's still kind of unclear how different factors determine which site is their site and to what extent they compete and what extent they. So, for example, if you look at the homeobox factors, box genes is like it tends to be more specific, I believe, especially when the binding motif itself is much smaller. Like you have a strong ACG here and a strong TC here, and very little else. Then these guys have to be there, otherwise you don't bind. It could be also partly an artifact of how all of these were basically in silico predictions from high throughput data. So these are chip-seq and these are chip-on-chip. They basically give you much larger sequences which are bound by the factor. and in there I go and look for a site using the canonical position weight matrix. There could be some artifacts induced from that too. But my impression is that yeast sites do tend to be stronger compared to multicellular organisms. Yeah. Yeah. It's not clear. So these factors, I actually haven't looked for homologs. Um, yes. Mm. It is a good question. So this ChIP-seq experiment, each of them was done for this factor, with this antibody. So, but it could be that some of these sites are binding sites for other factors too. And yes, so it's a good point. I should probably look at that. Yeah. So this is ENCODE data, and they have for something like 100 and something factors. Um, so whether they have done the experiment for the homolog is not clear. They might have, they might not have. Sorry, the promoted. Yes, yes, yes. But that should probably not affect the motif. You think? Yes. Right. So you're saying that the selection pressure will be more if it is regulating two genes. That's possible. Yes. It's perhaps even more common in yeast though, because the genes are just closer together. Yeah, these can be anywhere. Yeah. That's possible. Intron should be similar to proximal promoter, but well, I think enhancers, distal enhancers might be under less pressure, but it's it's something I should look at. It's true. Okay. Yeah. Yeah. Uh, it is usually A, but sorry, they are not competitive. This is a collection of aligned binding sites. Most of them had an A, and maybe 20% had a G. Whereas in this case, it shows the selective pressure here. It is basically the stationary distribution of the um, Felsenstein model as inferred from this collection of sites. And that stationary distribution again would tend to favor an A here, but sometimes a G. The compensatory part is, I guess, in the next slide. Supposing of this A and G here, I chose an A. Then, so actually this A and G are of roughly equal height, but if I choose an A, I see a slightly different pattern. If I choose a G, I see a slightly different pattern. Here, I guess the idea of compensatory here, it clearly prefers a G, but there is some sign of a C here. It turns out if I pick this G here, then I don't really care about the C that much. It's similar, but if I pick a C here, then I need to have these Cs. Right. It basically, one would think it affects the binding energy in some way. If I have a C here, it's detrimental to the binding energy, but then if I have these Cs, it brings it back. So I mentioned CTCF in the first slide, insulator binding protein. That's actually a very interesting case, and it's what spurred this idea. So that has complicated intra-site dependencies and a complicated evolutionary history, and so it seemed to raise many such questions. Okay, thanks.